Kommen wir nun zur Wissenschaft. Es ist mir eine besondere Freude, auch in diesem Jahr wieder, nun den ersten wissenschaftlichen Vortrag an dieser Fachtagung anzukündigen im Bereich Medizin. Auch in diesem Jahr dürfen wir Professor Bhupesh Prusty persönlich begrüßen, der sich die Zeit genommen hat und die weite Reise aus Riga, um uns heute seine Ergebnisse vorzustellen. Dafür, finde ich, sollten wir zunächst mal als Dankeschön herzlich applaudieren. You are coming to my side. Professor Prusty ist ein Experte auf dem Gebiet der Virusforschung und Immunologie. Er ist Professor am Institut der Mikrobiologie und Virologie an der Universität Riga und hat zuvor unter anderem am Universitätsklinikum Würzburg gearbeitet. An der Universität Heidelberg arbeitete er mit dem Nobelpreisträger Professor Harald Zuhausen zusammen. Sein Forschungsschwerpunkt liegt auf den Wechselwirkungen zwischen Viren und Wirtszellen, insbesondere im Kontext von chronischen Erkrankungen wie ME, CFS. Der Vortrag des Titels lautet Der passive Transfer von IgG-Immunkomplexen von postinfektiösen ME-CFS-Patienten verändert die mitochondriale Architektur, Funktion und induziert Entzündungen. Er wird zeigen, wie Antikörper aus dem Blut von ME-CFS-Erkrankten die Struktur und Funktion von Mitochondrien in den Zellen beeinflussen können und wie dies Entzündungsprozesse auslöst. Diese Erkenntnisse liefern wichtige Hinweise auf typische Symptome von ME-CFS, deren Ursachen und zelluläre Ebene, auf der sie entstehen. Wir freuen uns nun auf die Präsentation und begrüßen vielleicht mit nochmal einem Applaus Professor Prusty.
So, thank you once again for the invitation, Fatigatio. I'm so happy that I don't want to be the professor that you have invited, rather the Bhupesh, the guy who you know that he works in the field of ME/CFS. Yeah? So as you know, we are now working in Riga, and today my aim is not to tell about the research that we publish and you all probably can read, but to give you the big picture. The idea that why ME/CFS is still a not solved puzzle. Yeah, why we are facing so much of trouble to understand this disease. Yeah, so there are many hypotheses. There are many excellent hypotheses in this world, and everyone tried to solve this puzzle in their own way. Yeah, we have our own way of looking into the disease, and the two important things that we try to understand is the infection, yeah, what has infection to do with this disease, and the chronic nature of the disease. So what we believe is that the first important thing is that not every cell is sick. That means that we need to try to find out in millions of cells those one, two, or few cells which are actually sick. And why these few cells are communicating to all the other healthy cells to remain sick, this sickness response. Yeah? This is the chronicity. Yeah? So maybe these few cells which are sick because of infection or because of something else. So the ME/CFS patients are not different from healthy individuals that we believe. Yeah? You're sick, yes, something is going on inside, but as a whole organism, as a human being, you are not different than a healthy individual. What makes you different is when you come under stress. Yeah? That stress can be physical stress, it can be emotional stress, can be molecular stress, can be hormonal stress. Okay, every person, every patient is different. Yeah, what makes you different than a healthy person? That's how we try to look into this disease. I will go back to my dear friend and mentor Bob Navio's idea of cell danger response. He suggests that one single dysfunctional cell can pass on the message to hundreds of healthy cells in the surrounding, that understanding that, okay, I am sick, I am facing a danger, and this you should understand and be prepared. This communication between one cell which is sick and the hundreds of cells which are not sick is the key to understand this disease. So I'm going to today focus on two different aspects. One is what is there which is sent by this defective cell which is being recognized by the other cells. This is one of the aspects. Yeah? There can be many different things. And are these hundreds of cells are somehow different, that they are not able to respond the same way as a healthy cell will respond. Okay? So we will understand what is passed on and how these cells are responding. These are the two important aspects. To understand this, the first aspect is I would introduce you a very new technique that we have developed in our lab, which is called single-cell SLAMSEC. I will try to make you understand in the most simplified way, without going into the nuances of science. And why it is so important that that we focus on the ME/CFS patient, where everything is silent until you stress them. Yeah. So, just to give you an example, a very simple way. Think about it. I put you into a dark room. There is no electric bulb. Yeah, it's completely dark. And I ask you a question. Can you tell me whether the electric bulb is working or not? Can you answer it? No. Because it's dark, you have to switch it on. And then you can realize whether it is there or not, whether it's working or not. Yeah? The whole idea of this technique is that you switch on the response pathway inside the cell and then see whether the cell is able to respond it or not. So you need to switch on the bulb. Now this is a very simple cell, and there are many things going on inside the cells, and there are, in any given time, you have a lot of mRNA inside the cells, the genes, the transcripts. When this cell gets an infection, the infection is taken up by the cell. Its cell understands there is a virus or the bacteria or fungus, something is coming in, and it changes many pathways and genes, and new RNAs are produced. Yeah? Now think about it. If I have an ink, just to give you a layman idea, which basically can paint all those new RNAs which are formed only when the cell is seeing the infection. Not before that. When the virus comes in, something is changing, and these new things are all painted by this paint. Yeah? The idea of this, so what we do is that we basically capture these new response genes by using RNA sequencing, and that tells us how these cells are responding to that stress. Again, I won't go into the details of this technical process, how we do it. It's a very expensive process. Just give you an example why this process is so informative. Now, we took almost 3 years now to develop this technique in our lab. It was initially developed by Stefan Amrist's lab in Vienna to look into bulk, like hundreds of thousands of cells together. Well, we went ahead and we developed this technique to look into individual cells, T cells, B cells, monocytes, NK cells. They're all different. Yeah. So we did this experiment. This is a pilot experiment. Today I'm not going to go into the details of the result. It's too early. But I will tell you why it is so powerful. So what we did is that we took blood from healthy controls and ME/CFS patients. And we took these blood cells and we kept them like without giving any stress. In one way, we gave the stress of mimicking a virus infection. what will happen if a ME/CFS immune cell sees a virus, flu, or something else. Same way, if a messenger molecule like extracellular ATP is given, so what will happen? And then compare how the healthy cells behaving to the ME/CFS patients. And then we use the single cell sequencing approach. Now, it's very interesting is that I told you that you need to switch on the bulb to see the effect. Now, look here. This is how the results look like. And so here, these are hundreds of genes under a very normal condition. Yeah, so some genes are expressed in, so each column is one cell. So you can see that some of the genes here are randomly expressed in some of the cells. But there are a lot of genes which do not express under normal conditions. So your cell doesn't do everything when it is resting. Yeah. Now, when we put the poly-IC, it's a chemical which basically tells the cell that there is an infection going on. Yeah, in a very simple way. So you see that all those genes which are usually silent, they start to pop up or start to express. Yeah. The same is also here. You see that a lot of genes which are very mildly expressed. The moment you put extracellular ATP, they start to be on. Yeah. This you do not capture by all the published techniques so far which you see, because under normal conditions you don't see them. And we are trying to see them, trying to take the blood cells from the ME/CFS patients and healthy controls and give them many different type of stresses, okay, like an infection, like a antibody which can produce mitochondrial dysfunction, yeah, and trying to compare how your or patient's blood cells behave in comparison to the healthy controls. Now, why is it so interesting? Look into this result, which is basically a broad representation of what we get. So on the left side, you have healthy cells. So this is not one person. This is a combination of many healthy, blood cells from many different individuals. And here is from ME/CFS patients. Now, how the B cells responded, how monocytes responded, how NK cells, natural killer cells responded, how T cells responded. So if you see in the blue, the poly-IC treatment. The red is the extracellular ATP treatment. Now, what we try to understand that a ME/CFS patient is sick and sick all the time, and probably will do the same thing under every condition. No, it's not the same. Look here. If we put the poly-IC like an infection, there are 35 genes which are changed within 2 hours of treatment. They are not exactly the same when the same B cells are exposed to extracellular ATP. There is zero overlap here. That means the same B cell, if it sees an infection, will behave differently than if it is under a different stress. Okay, so every stress is different, and we have to understand this individually. So this slide basically shows you that there is no overlap between two different stressors which is being tested. But if you look into the ME/CFS versus healthy controls, you see that if the B cells from healthy individuals are given an infection, there are 35 genes which are changed within the first 2 hours. But when it comes to the ME/CFS patients, the response is not there. Only 5 genes changes. Yeah? That means that the ME/CFS patient B cells are not healthy enough or equally good enough to respond to an infection which is coming in. Yeah? On the other hand, if we try to look into the, for example, the NK cells, you see that the healthy, the extracellular ATP treatment in healthy controls, they change 71 genes within first 2 hours, whereas this here in ME/CFS patients is only 26. Yeah? So the whole idea is that by differentiating individual cells, hundreds and thousands of cells from healthy individuals versus ME/CFS patients, we are able to see the exact difference between cells, between types of cells, and between different type of stress. Yeah, so you can ask question that, okay, we were discussing yesterday that I can swim 2 hours, nothing happens. But if I take a little bit of weight, then the cross happens. Yeah. So what is the difference? What exactly is the difference between these two stress? It's not exactly the same thing. Yeah. So this process is still under development, and we have been lucky enough that the Armour Foundation from US is spending actually millions in developing this technique. It normally costs around roughly 20,000 euro to test individual person's blood sample in this process. But we have been decreasing the cost, and now we are in the range of around 5,000, and hopefully we can bring it down further. So what we learned from this process is that the ME/CFS patients are not much different from healthy individuals until you actually give them the stress, okay? So different cells behave differently to stress, and ME/CFS cells, they activate many signaling pathways that actually contribute to the chronic aspect of the disease. Yeah, we heard about, we know that reactive oxygen species or the oxidative stress is one of the very important aspect of ME/CFS, and this process actually shows how the oxidative stress is built in ME/CFS cells in comparison to the healthy individuals. Now, again, it's too early to conclude anything, but the whole experiment supports the idea that hypometabolism is one of the key features in ME/CFS, which we know from many years. But it contradicts one of the very well-known idea that ME/CFS patients have an increased immune system, which basically we also suggest that in a couple of years back. But this is contradicted by these experiments. Okay, it's still underway. We will see how things develop from here. Now, this is all about how the cells respond, the healthy cells respond to a sick cell. But what is that process that is actually passing on from the sick cell to the healthy cells? This can be many factors which basically are there in the serum, the complement activation process. It can be constant stress on mitochondrial morphology that can pass this information through immunoglobulins, and many other factors, for example, inflammation because of the cell death. It can be small RNAs by viruses, it can be mitochondrial DNA, it can be proteins or RNA loaded into exosomes which are secreted outside the cells, and it can be also many different type of stimulatory factors secreted by eosinophils or mast cells in the form of the activation. So we know from many years that if you take the serum from ME/CFS patients and put a healthy cell into the serum, you can actually mimic many of the phenotypes that you see in ME/CFS patients. But we did not know so far what exactly is this factor. As I said, it can be many different factors. There is no single factor. We focused on immunoglobulins. This is the project which BMBF supported us, and we looked into how immunoglobulins can cause mitochondrial dysfunction, inflammation, and immune modulation. So again, a very simple way of looking into the problem without complicating the experiments. So what we did is that, first of all, we isolated immunoglobulins from hundreds of individuals, ME/CFS patients. COVID-induced ME/CFS patients, like the very fresh 2 years into the development of ME/CFS, we took multiple sclerosis patients as controls and healthy individuals, yeah? And first question that we asked is that, can these antibodies, it's basically combination of IgG, a little bit of IgM, IgE, IgA, basically. Can these immunoglobulins actually enter into the cells? Because this is a big question in the field of endocrinology also. You can have effects of antibodies by being the antibody outside also. But if at all the antibody can enter into the cells. So we exposed, for example, here, 2 different types of primary cells. The human umbilical vein endothelial cells, so these are primary endothelial cells, and human foreskin fibroblasts, yeah? So 2 completely different type of primary cells, and we put the antibody on these cells, okay? And then try to see if the antibody is actually entering into the cells. And if you see on the right side, there is no antibody entering into the fibroblast. But at the same time, the same antibody nicely enter into the endothelial cells. That means your blood circulation can have many different type of antibodies, and these antibody will affect differently to different type of cells. You cannot generalize this whole process. Yeah. So you can also see that if you put the antibody into cells, you can actually stand them and see them inside the cells. The next question we asked is that how long the antibody actually can survive inside the cells once it enters into the cells, okay? The recycling time of these antibodies. And you can see here that 16-hour post-entry, 36-hour, 48 hours, 72 hours. You can see that by 48 hours, you still can see a lot of antibody inside the cell. But by 72 hours, these antibodies are usually removed out of the cells. They are recycled, okay? So they are broken apart, and everything is recycled, basically. And one of the interesting aspects that came out is that antibody normally have two fragments, the Fab fragment and the FC fragment. You can see that a antibody, the FC fragment are much more stable inside the cell in comparison to the Fab fragment. Now, when we put these antibodies into endothelial cells, we realize that a strong mitochondrial fragmentation is happening in these cells. Yeah? So mitochondrial fragmentation means the long elongated mitochondria are broken down into smaller mitochondria. I want to warn you here is that mitochondrial fragmentation is not always bad. Your mitochondria has to fragment to be recycled. So for being healthy, you need to have a constant process of fusion and fragmentation. If it goes in one way, then it is bad. Yeah? So we saw that the mitochondria is fragmenting, and this fragmentation is actually very strong in ME/CFS patients. In comparison to the freshly COVID-induced ME/CFS patients over here, you can see, but it's not a uniform process. You can see that a lot of overlap is happening between the healthy individuals and many of the ME/CFS patients. But what is very interesting is that a subgroup of patients over here, and these are mostly women, yeah, who very strongly responds to mitochondrial fragmentation when the antibody from ME/CFS patients is introduced into healthy cells. So as I mentioned briefly here that the immunoglobulins has two parts, the Fab fragment and the FC fragment. So we asked the question, okay, we are putting the antibody into a cell and concluding that the antibody is causing the mitochondrial fragmentation. How to control it? How we know that it is the antibody which is doing the job, nothing else? So we basically divided or digested this antibody into two fragments and separated them, yeah? And then we put them into the same cells. What we saw is that there is more or less no significant mitochondrial fragmentation happening if we divide the antibodies into the two fragments. Yeah? You only see it when the antibody is intact. So next question came, okay, the mitochondrial fragments, what does it do to the cell, to the mitochondria, to the ATP generation process? Yeah? So this is all about looking into the energetics process using seahorse bioanalyzer assay. Again, let's not go into the details of technical aspect. But one thing which came very clearly is that the ME/CFS patients, they have an increased spare respiratory capacity when the antibody enters into the cell. What does it mean? Spare respiratory capacity is not a negative thing. It's actually good. The more healthier the cell is, or the more responsive the cell is, it will have more spare respiratory capacity. It means that when you need more ATP, you have the possibility to produce the ATP. It's completely contradictory to what we think. We think that ME/CFS patients are sick, they cannot produce ATP. But here it came out that actually the ME/CFS patients, they are tuned to be more responsive to stress. And they can actually produce more amount of ATP. This is not a new thing. This result is actually published by Pluge and Mella long time back by putting the serum. We for the first time saw that actually antibody alone can also do the same thing, exactly the same thing. But what is very important here to understand is that You can respond to stress, but if you keep responding to stress for year after year after year, then the whole process gets saturated, okay? And that creates the defective process. We actually showed that, so here we showed that if you divide the, again, digest the whole antibody into the Fab fragment and Fc fragment, the process becomes completely different. You look into this picture itself, you see that the red is the ME/CFS patients. When the Fc fragment is separated from the Fab fragment, yeah, you go back to the previous picture result where I showed that after 48 hours, you don't see the whole antibody inside the cell. It is being digested and only the Fc fragment is still surviving inside the cell. And if that is the condition, then you see that the cell becomes hypometabolic. That means alone the Fc fragment of ME/CFS patients can cause the mitochondria or the whole cell to go into a process of producing less ATP. Yeah? And this is not because of mitochondria, this is because of the process of defectiveness in glycolysis. So far we have been thinking that mitochondria is doing everything. The picture is changing here. Actually, mitochondria is communicating with the cell. So take an example. If you are going for a quick run, yeah, when you are starting the run, all the energy is produced within the mitochondria. But the moment this mitochondria, this ATP is exhausted, and you need more amount of ATP, it's automatic switch to the glycolysis. Yeah, it's not bad, but that process is being defective over here. Yeah. Even though the result is not significant, here also it shows that the Fc fragment alone can cause a significant decrease in glycolytic ATP production from ME/CFS patients in compared to the healthy individuals. And this is so significant that you can actually see the same pattern in the total ATP. Yeah? When you look into the mitochondrial ATP, you don't see much difference, even if there is a sort of a trend, but it's not statistically significant, but close enough that we can trust this data. Yeah? The next question that we ask is that, okay, the mitochondria is, the immunoglobulins are causing the mitochondria to respond to produce different type of ATP. Does it have anything to do with the inflammation? So again, we do interesting way of understanding this process. We isolate immunoglobulin from the patients, take healthy PBMCs, not the culture cells, the PBMCs, and expose these immunoglobulins to these PBMCs and let them respond to it by producing cytokines, yeah, pro-inflammatory, anti-inflammatory cytokines, and then measure it. So you can see here is that a large number of ME/CFS patients, it's not a uniform process, they actually, their immunoglobulins actually induce cytokines like IL-1 beta, interferon gamma, TNF alpha, IL-6. Yeah, these are all pro-inflammatory cytokines, which are very, we know that they are high in ME/CFS patients. Again, when we talk about these results, we are criticized very often that, okay, you don't have a statistical significance here. Yeah? We are not going to solve this puzzle overnight, and we are not going to create a solution. But what it tells, the basic concept, how things are working inside the cells. Yeah, not every patient is same. Yes, we have hundreds of patients. Definitely, not all the patients are severe patients. Not all the patients behave the same way. But at least we are able to see the visible effects of an individual factor in this process. Yeah. What was interesting here is that if we compare the different type of cytokines being produced by these immunoglobulins, what we see is that the long-term ME/CFS patients, sick for 15 years, 20 years, their immunoglobulins are much more different than the freshly induced ME/CFS patients, the COVID-induced ME/CFS patients per se. The COVID-induced ME/CFS patients are much similar to multiple sclerosis. Yeah, I don't say that they are multiple sclerosis, but when we look into this particular process, they are much closer, yeah? Then, again, the question came, okay, we are taking the immunoglobulins, putting into the cells. Immunoglobulins can have also antigens. There are a lot of protein binds to the immunoglobulins. So what are those? Can we see some differences? So we did a mass spec analysis of all those samples, which we tested in the cells. Now, this is a PCA analysis over here. Okay, PCA analysis is basically, you don't look into 1, 2, 3, your favorite protein. You just take the whole data. as a bunch and try to see on the basis of this data, can I separate healthy controls from ME/CFS patients or from COVID-induced ME/CFS patients? So if you look here, red are the healthy controls, and you can see that they all, in a three-dimensional space, they all occupy a different place than the three disease group that we studied here, yeah? And even though the three disease groups are pretty close to each other, it's very exciting to see. It's not really the black and white picture, but it is very exciting to see that all those green ME/CFS, the long-term ME/CFS patients, they come towards a different side of this three-dimensional space in comparison to the COVID-induced ME/CFS and multiple sclerosis patients, which are much more overlapping with them. That means that these proteins or antigens bound to the immunoglobulins are contributing to the process of disease development. Again, a lot of data, a lot of protein to analyze, so we can see that some of the proteins or antigens which are altered or upregulated or downregulated, they represent all the three disease groups, but there are certain proteins which are very specifically changed in ME/CFS patients or COVID-induced ME/CFS patients. I just presented one data over here. The KLKB1, it's a serine protease precursor, and it's responsible for converting the plasma kallikrein into proteins which are responsible for blood coagulation, fibrinolysis, inflammation, and blood pressure regulation. And we actually validate it in a larger cohort and show that actually there is a difference in the amount of this KLKB1 on your immunoglobulins. Another very interesting protein comes off the SPEG over here. That's a striated protein, striated muscle fiber protein, which is responsible for the successful working of muscle fibers, and that is very specifically altered in ME/CFS patients. Okay, when I say altered, it's inside the immune complex. Yeah. Again, let's not go into our individual favorite proteins. Try to generalize the results. What we see is that there is one pathway, so this is gene set enrichment analysis. There is one pathway which defines or which is changed significantly in ME/CFS patient versus healthy control is the extracellular matrix organization. Okay, it's not because of one, two, it's multiple different things changes, and you will be probably be excited to see that the recent paper which came out from the Jan Lipkin's lab, they also saw that one of the major things that they see ME/CFS patients is the extracellular matrix. Now, what is extracellular matrix? Is basically the area within your tissues within your body that holds your tissue intact into a three-dimensional space, basically. So why it's very interesting, you probably know that we have one paper in the process of revision now where we show that one very specific protein, fibronectin, is changed dramatically in ME/CFS patients. So you can see here, this is a cohort of patients from Germany, and you can see that the amount of fibronectin in the serum is significantly high in ME/CFS patients. If you look into another cohort, completely independent cohort of ME/CFS patients in Sweden, we see exactly the same trend that these fibronectin is extremely high in the serum of ME/CFS patients. Now, what is fibronectin? It's one of the most important component of your extracellular matrix. Yeah, and again, it's coming back to the same story. Is that the extracellular matrix basically responsible for wound healing, responsible for mast cell activation, the process of inflammation, cell-to-cell communication. These are all processes which is affected in ME/CFS. Now, the big question is that whether the long-term ME/CFS patients are exactly the same as COVID-induced ME/CFS patients. Yeah, I would be careful to make any conclusion, but we do see differences. Okay, and I personally believe that these differences are because the patient who are just converted or just started to develop the ME/CFS, they are going through the very early stage of development of the disease, the most ideal stage where you should find a marker for them. Yeah. Those patients are different from the long-term ME/CFS patient who has crossed the barrier and gone to the chronic part of the disease. So what is the big difference here is that The ME/CFS patient have a different response to bacterial infections in comparison to the COVID-induced patient. But as we hear from many of our colleagues, probably today also you will hear from some of our other colleagues, that the long COVID patients or the COVID-induced ME/CFS patients have defective clotting process, and the blood hemostasis is different, is exactly what we see over here in a completely different approach. Yeah? So coming to the last slide, summary, what we saw here is that yes, the ME/CFS patient are different from healthy individuals at the cellular level, but you do not see the difference until unless you really ask them the question, can you do this? Yeah, this response to stress is the defective part, not overall the cell. And one of the mechanisms by which the defective cell pass on this information is probably through immunoglobulins. And what we see is that the immunoglobulins enter into the cells. They are probably digested, recycled. But in ME/CFS patients, once this process happens, these Fc receptors, they start to affect the process of the ATP generation. So they push into the hypometabolic phenotype. So last, I'm very thankful to many of the funding agencies which basically keep us alive in this field for last 14, 15 years. We just finished our lab in Würzburg. Some of the colleagues, they are moving now to Riga, so we are expanding now in Riga. This year itself, we have accumulated more than €1 million of funding for ME/CFS research, sitting in Riga alone. So we are doing good. There are a lot of different agencies who have been trusting our work and funding, particularly the philanthropic organizations who are giving us the free hand to think freely and to do different aspects. I would like to mention Fatigatio. Your help is absolutely phenomenal. The way you helped my student Stefan to finish his work in Germany, I mean, it's beyond words to appreciate these timely help. So thank you very much, and I hope that I could tell you that we are with you. So we think about you. We try to solve this problem very sincerely, and trying to get to the best way that we can solve this problem, rather to put it this way, yeah? Not just simply giving you 10 different papers saying that, okay, this happens, which doesn't yield to anything at all. Rather to bring it all together to one place where you can actually think, model, and see, okay, what is going on here? Yeah? That's what the whole aim is all about. Thank you very much.
Yes. So, thank you very much.
I took 5 minutes extra, sorry.
Thank you very much for the presentation. Professor Brinkmann, Herr Brinkmann, laufen Sie nicht weg, Herr Brinkmeier. So schnell konnte ich gar nicht reagieren. My thank you today is the next year. Please come again in the next year. This is for you.
Thank you. So I guess there is no time for questions or something like that. But I'm here at least till the lunchtime, and I'm really, really excited, happy to talk to you. And maybe sometimes come to Riga. It's a beautiful place. It's
wow,
a beautiful
money?
place. No, it's cheap. Yeah. If you're going on holiday in France, Germany, Netherlands, Belgium, you can probably do it better in Riga in half of the money.
Oh, no problem, no problem. So, thank you.
And yes, we are building a big team. We applied this year already 2 big European grants, crossing fingers that something will work out. So please, yeah, thank you.
I say thank you. Applaus, oder?